Hi, everyone. I'm Polly Chen from Cancer Science Institute of Singapore, National University of Singapore. I'm honored to host today's seminar. As you may know, the next RNA Society conference will be happening in Singapore. Actually, RNA biology is becoming a priority area in Singapore. We have a very vibrant RNA community here. So today we will be having two great scientists from our very active RNA community to share their exciting research with us. Our first speaker today is Professor Dan Tannen from Cancer Science Institute of Singapore, NUS. Dan is a leader in gene regulation in both normal differentiation and cancer with a focus on leukemia and other type of solid tumor, for example, lung cancer and liver cancer. He was the lead PI for a 25 million program grant from Ministry of Education Singapore, which focus on the RNA biology in cancer. Today, I believe he will be sharing some very interesting findings on how transcription factors, which are known to bind to the DNA elements, act as RNA binding protein to regulate gene expression and the cellular processes. If you have any questions, please use the Q&A function to raise your question. I will read out the question during the Q&A session. Without further ado, please join me to welcome Dan. Over to you, Dan. Thank you.
Okay, I hope everyone can hear me.
Yes.
This is going. Okay, and get away to the pointer. Okay, so I'd really like to, first of all, thank the RNA Collaborative. I think it's a great effort. It's around the world. It's just wonderful talks all the time. Just love it. Also, thanks, Polly, for inviting me to talk here. Today, the title that I had before is not exactly what I'm going to talk about. Transcription factors. Transcription factors are sequence-specific DNA binding proteins, right? No, they're RNA binding proteins. So once upon a time, my lab did study these as DNA binding proteins. But recently, late last year, we published a paper in Blood. I'm just going to briefly mention this. Let me get this thing. And that was in Blood, and it was really the work of a really smart senior fellow in my lab, Chen, who now has his own empire in the University of Virginia. And basically, he described a long non-coding RNA called loop that emanates from the Pew enhancer, which is called the upstream regulatory element at 17 kilobases upstream of the promoter. And it's expressed from this enhancer region. It's expressed in a myeloid-specific manner similar to that of Pew. And RUNX binds the loop and is recruited to RUNX DNA binding sites in the Pew promoter enhancer by the RNA. RUNX1 DNA binding to the promoter and enhancer induces chromatin interaction between the promoter and enhancer, and that's to messenger RNA expression, and that's how you get to expression. Basically, the RNA pulls in RUNX, RUNX binds to the enhancer DNA, you get chromatin interaction, you get to expression, and in one form of acute myeloid leukemia involves a translocation that produces a RUNX fusion and actually inhibits loop expression, and you lose Pew expression. And what this demonstrates is the role of transcription factor RNA binding to induce recruitment and DNA binding to promoter and enhancers. And it's also interesting, as Bon pointed out, it's a mechanism of how a non-specific transcription factor like RUNX can induce a target gene like Pew in a myeloid-specific manner, because the loop RNA is expressed in a myeloid-specific manner. So it's kind of a cool, interesting mechanism how a non-specific factor might become specific in terms of binding to RNA. So for many years, my lab has worked on this transcription factor. It's the famous one. It's the famous VZIP protein. This is a crystal structure of CBP Alpha that Steve McKnight described in 1989, ancient times, and the famous leucine zipper. And we did a lot of stuff with this. It's very important in acute myeloid leukemia as well. But that's all boring now. what's interesting, of course, is RNA binding. And so, question that we wanted to ask: does CBP alpha interact with RNA? And it turns out that we did RIP sequencing, and CBP alpha interacts with RNA in mostly intronic sites. This is using 1% formaldehyde cross-linking in some leukemic lines. And we picked as a target to study further PEW itself. So there's a site in intron 4 of PEW in the RNA, in the primary transcript, that binds to CBP alpha quite well. And this just shows RIP PCR, just showing good binding to intron 4, and as a control we use intron 2, in which it doesn't bind very well. And then in addition to the formaldehyde cross-linking, we also, I can't get this to move. Okay, there we go. We actually use CLIP, which is UV cross-linking, to also validate that PEW could bind to, that CBP alpha could bind to this side in intron 4. And there's another gene that we looked at again, where it binds better to intron 2 in that particular gene. So what's one interesting thing about CBP alpha RNA binding, a few years ago we described how DNMT1 could bind to RNA and regulate DNA methylation. And we think that the binding there involves shape. We really couldn't figure out a good sequence for it to bind. But we think in the case of CBP alpha, there are some motifs that it likes. They're mainly, as you could say, CG rich. These are 3 of the motifs that it likes to bind to. And if you look in PEW intron 4, you see this, these motifs. And again, in CTBP1, another target, again, a lot of these motifs. So we think that there's actually some sequence specificity to the binding to this site. Now, here came the big surprise to me, because I predicted, this is a nucleic acid binding protein. This is the sort of the linear structure of CBP alpha. It has transactivation domains and the amino terminus, which I'm really wondering, are these things transactivation domains? They may be RNA binding domains. And here's the famous B zip and the C terminus. And I really thought it would be the B zip that binds to RNA, just like it binds to DNA. Okay, binds to one nucleic acid, will bind to the other. So here's the B zip fragment doesn't bind at all. This is a REMSA assay using P32. We're very old-fashioned. And again, in this CTBP1 site, again, the B zip doesn't bind at all, whereas the folate protein binds quite well. And here's some of the controls. I won't have time to go over all the data, but we don't see a shift when we use a double-stranded DNA probe from the same sequences. And this B-zip does bind very strongly to a proper DNA binding site in the PEW enhancer. So we really think it's a specific binding not mediated by the B-zip at all. And that, to me, I was so surprised. By the way, here are some probes from CTBP1 where we have a linear probe that does bind, and then we made some modifications to sort of induce a secondary structure that doesn't like to bind. Okay, we made a whole bunch of purified small deletions in the PEW to try to define, what sequences actually mediate binding in. We got really smarter than my own brain. We used AlphaFold 2 to predict DNA RNA binding regions, and then we tried to validate it using all these different fragments. And amazingly enough, we actually see something consistent, that the areas that were predicted to bind to RNA actually did. Again, in REMSA with P32, there are 2 regions, relatively small regions, amino acid about 30 from 70 to 97 and 159 to 182. And the blue lines, you can see they bind very nicely. And a fragment that has the B-zip does not bind to RNA at all. And again, these 2 regions here of about 30 and 20 amino acids bind quite well. And again, we didn't see any shift observed when we used double-stranded DNA probes from the same sequence. So we really think it's RNA binding and not DNA binding. Then we looked at affinity, and we used this method here, in which we use increasing amounts of protein, and then using an RNA probe from intron 4, and we compare a DNA probe from the PEW enhancer. And when we do the calculations, we actually see pretty similar affinity binding with RNA and DNA. So, and similar results using ITC. So we think the RNA binding is actually has fairly high affinity, at least in vitro. So Now we get to the question is, well, so what? So my former postdoctoral fellow, Rob, who now has his empire at the University of Alabama, Birmingham, he always says, why should I care? So does it have any function? And so to do this, we devised a scheme to actually look in real cells in the mouse. And so to do this, we took a conditional knockout mouse for CBP alpha that we described, I think, in 2004 for the first time, looking at its DNA binding effects. And then we induced deletion, so we have a knockout mouse for CBP alpha, and then we put in CBP either wild type or a double deletion mutant that contains those two small amino acid domains that bind to RNA. Now, you can't put CBP alpha in constitutively because when you do that, the cells stop dividing and you won't get any readout. Actually, we published a couple papers. Well, Scott Kogan published a couple papers. We were just collaborators in the 2000s actually showing that you have to use an inducible CBP if you want to see anything. And so we put in this construct and we wait, and then we put in, we induce the construct using estradiol. And then we can look at the readout. And when we do this in culture, you can see the wild type vector. Vector alone does nothing. The wild type gives you these nice macrophage cells, and it gives you induction of macrophage markers like C11D and F480. Whereas when you use the double deletion, you don't see anything that looks like a macrophage, and you don't see induction of these markers. Well, that's in culture. What about in real cells? And so we did the same kind of protocol. We induced knockouts, and then we put in the inducible CBP wild type mutant constructs, and then we transplanted into congenic mice, wait for some reconstitution, then induce the CBP alpha wild type mutant constructs and look at what happens by FACS. And I'm just going to summarize the major defects that we saw. So in wild type cells, you see induction of macrophages. These are macrophage markers, whereas in the double mutants, you actually do not see this population at all. This population is more of a monocytic cell type, not the macrophage. And again, using a different marker, you see the same effect. And this is bone marrow. We saw similar results when we look at splenic cells. So what's going on here? And so we looked at sort of the mechanism of what we think is going on here. We think that these RNA binding deletion mutants, they don't bind as well to Pu enhancer because they're not recruited there. Then you have decreased Pu messenger RNA, and Pu is very important for macrophage gene expression and differentiation. So we think that's the mechanism. Here's some of the data. So this is using primary cells from bone marrow and spleen. And these are 3 CBP alpha binding sites in the Pu enhancer. And you can see big loss of binding when you use the RNA binding mutants. And the interesting thing is that if you look at other CBP targets, like CBP epsilon or GCSF receptor, that CBP alpha binds just fine to those. So that's why we think it's a local RNA which is recruited to the enhancer, because if you look at more distal sites, where we don't think this mechanism is going on, you don't see any effect. You only see it in cysts in that locus. As a result, you see decreased Pu messenger RNA in the double mutants and decreased macrophage gene expression. Now, one surprise, at least to me, again, was that these double binding deletion mutants show no effect on neutrophil differentiation. So if we look in the spleen and bone marrow, here's the wild type. These are neutrophil markers, and the double deletions really look just like the wild type. The control, if you look at CBP knockouts, you don't make these granulocytic cells. So we're wondering maybe high Pu levels are not needed for neutrophil differentiation. It is interesting though, sort of the dogma is that, CBP alpha is up in neutrophils and Pu is up in monocytes and macrophages. Well, it actually turns out if you look at real data, both in mice and humans, we and others have published, Niels Beauregard as well, that actually Pu is expressed at fairly high levels in neutrophils, but clearly it doesn't seem to be needed for neutrophil differentiation, maybe for function. So here's sort of our model. So in the wild type, basically what happens, you have CBP alpha, and it's recruited by these RNAs in intron 4. That recruits local concentration of CBP alpha. You get binding to the enhancer, you get promoter-enhancer interactions, you get few expression, you make neutrophils and macrophages. But if you delete the RNA binding function, then you don't recruit as much CBP alpha, you don't get recruitment to the binding sites in the few enhancer, you don't get promoter-enhancer interactions. Excuse me, you don't make macrophages, but interesting enough, you do make neutrophils. We're not quite sure how that is working. It's quite interesting to me. So to summarize, CBP alpha likes to bind to RNAs and introns. The big surprise, the binding activity is not in the C terminus. Instead, it's in two relatively small sequences in the so-called transactivation domain. Seems to have a sequence selectivity, not like DNMT1. It can bind to few intron 4 RNA, it recruits CBP alpha to bind to the DNA binding sites in the few enhancer, and augments promoter enhancer interactions in few mRNA expression. If you delete these binding domains in vivo, you lose binding to the few enhancer, decrease few messenger, decrease macrophage gene expression. And you really don't see macrophage developing in the mice. And this is interesting again because it provides a potential mechanism of autoregulation of a target gene. So the more primary transcript you might make, you might recruit more CBP alpha to bind to that intron RNA and then you get more enhancer activity because then you get more binding to the enhancer. So it's an interesting potential mechanism. Now, what about Q? Okay, does it bind to RNA? Well, the answer, of course, is it does. And so here's some eClip that we did in collaboration with Gene Yo's lab, and some of the genes that seem to bind to Q, and in particular, these are a number of 3' and translator regions. If you look at where Q seems to bind, it seems to like introns, 3' UTs, and intergenic regions. And so we focused on the C-Jun 3' untranslator region as a probe, did some REMSs, and it's very much like CBP alpha. It appears to bind to RNA, but again, a big surprise, it's not through the C terminal S DNA binding domain, as I would have predicted. Once again, I'm wrong. But instead, actually, we've localized it to a single 10 amino acid peptide in the immunoterminal, again, so-called transactivation domain. I'm beginning to call these things RNA binding domains, not transactivation domains. And then here's a whole bunch of different fragments we made. And again, the S domain, which is shown here in the red, it doesn't bind at all. That's the DNA binding domain. This is a small peptide of about 20, roughly 20 amino acids. And if you make even more smaller, you can get it down to about 10 amino acids that'll bind pretty well. To an RNA probe, you don't get any binding to a DNA probe. So that's as far as we've gotten with Pew, but we're beginning to think that this may be a common factor in many so-called transcription factors, which are sequence-specific DNA binding proteins. So we've published on RUNX. CBP alpha, I showed you quite a bit of the data, and we're just beginning to look at Pew. Like CBP alpha, it's in the amino-terminus, it's not in the DNA binding element, and we really are just starting to do functional studies. So I think I have a couple more minutes. I just want to show 1 or 2 slides on some of the other things we're doing on RNA in our labs. We're interested in weird, I call them weird because you never can predict what RNA is going to do. Long non-coding RNAs, we talked about Beer's DNMT1 interacting RNAs a few years ago, regulating methylation. The 2 A's, Alex, who works with me, and Annalisa, who has her own empire now at Harvard. They have a new class of RNAs, which they call SPEARs, which come up very early following double thymidine blocks, so they're early S phase RNAs. They come up before the DNMT1 interacting RNAs and before the messenger. This is CBP alpha. We've done a lot of work as MYC is a target gene. Again, the sphere comes up first, the messenger later. We think they do a lot. We think they regulate messenger RNA transcription, epigenetic memory, DNA replication, and actually Alex has some really crazy data about reversing DNA trinucleotide repeat expansions using RNA, presumably because it affects replication. Meanwhile, we submit, reject, submit, reject. Oh, well. We got another rejection on this work today, just to congratulate myself. One other thing that we've been doing with RNA is a former PhD student, Yanjing Liu, we call Cupcake. She's now a postdoc at the Broad Institute at Harvard and MIT. She came and worked in Boston for a summer. She came up with the idea that something we call CRISPR-Dir. Basically, this is Feng Zhang's structure of a CRISPR guide RNA. Yanjing put in 2 sequences from this CBP alpha non-coding RNA that interacts with DNMT1 that we described in our Nature paper. And she makes this RNA here that has these 2 RNA binding loops in these 2 loops that are not needed for guide function. And we think it's a really good way to get gene selective demethylation. We've actually used it quite a bit. We use it to identify a regulatory element in the cell exon-intron border. And that's contributed to actually 3 publications working with Leilei at Brigham. Here's one from Cancer Research. One actually a really interesting paper where we show a pseudogene of SALL4 can activate in liver cancer working through that element. And then finally, a recent New England Journal paper from primarily from Lee's lab, in which we demonstrate, again, through this element in real patients that demethylation agents used in myelodysplastic syndrome and AML can actually induce SALL4. And if it does, through this element that we identified using CRISPR-Dir, they go on to a very bad outcome. We identified a similar element in P16, again in a similar region, regulates P16 silencing to a CTCF binding site. And there are thousands of genes that have these elements. They're about 4 nucleosomes in size. Work of Yanjing and my bioinformatician Mahmoud, who's really brilliant. They basically identified thousands of these mesas, and we've validated about 6 or 8 of them. Again, this kind of unique DNA methylation regulatory element. So I am done with the science. I just want to give a shout out to all my funding, which has funded these studies. Well, oops. I'm happy to say I have no funding for anything I spoke about. So I'm passing the hat. You can donate by Zoom, no matter how small. And it is Yom Kippur, the Day of Atonement, in which we all should think about our sins for the past year and vow to do better next year. And so those of you who trashed all my grants on this stuff last year, how about atoning for your sins and be nicer to me this coming year? Anyway, before I take questions from you, I'd like to ask a question of the audience, and you have to tell me, what is Yanzhou doing in this picture? Well, it's very obvious. I know none of you will get it unless you've worked at the Cambridge, the Cancer Science Institute in Singapore. It's very obvious what he's doing. If I can get my animation to work. He is winning the nose pull contest at the annual CSI retreat, of course. So for further explanation of this, I can talk to you afterwards, and I'll be happy to take questions.
Thanks, Dan, again. So, so you have 3 questions then. So the first question, or actually more, really asking, sorry if I pronounce your name wrongly. Have you tried to overexpress only the RNA binding motif to see if CBP alpha still binds?
Yeah, we actually have done that in cell lines, and it inhibits. What we can do is we can put in different sequences and test it, and we can inhibit binding by overexpressing it. I think we did it in HL60 cells, as I recall. I think that's what the question was.
Okay. Xue Bing asking, is there a chromatin loop between Q1 intron 4 and the promoter?
Yeah, it's a good question. We really haven't seen it when we've done studies of the chromatin interactions, not recently, but when we were first defining the Q enhancer, we only saw promoter and the enhancer interactions. We didn't see that.
Okay. Another question is CBP alpha binds to intron. So any evidence that CBP alpha change RNA splicing and further cause differentiate? Yeah.
Yeah, it's a really good question. We really haven't looked at it very much. We try to avoid the cytoplasm. Don't ask me why. I mean, so we haven't looked, but it's a good question. And, you know, I don't mean to imply that the binding activity of these proteins only works in the nucleus. It's because, don't ask me why. There's no good reason. I like to focus in the nucleus. I ignore everything in the cytoplasm. I have no idea why, but they easily could affect cytoplasmic events. I mean, I know splicing does happen in the nucleus. It's probably easier to see after what the results are later on. We really haven't looked at that question. It's a good question, and it's very possible that the RNA binding could have other functions.
Okay. Then there's another question from Yuping. Can I know the shortest RNA sequences for protein binding?
Yeah, I think I showed on one of the slides. I mean, they're pretty short. They're like typical, you know, EMSA kind of sequences, about 20 mers. I showed the sequence. I mean, I can go back and, or I can count it and tell you. It's no secret. It's just like a typical EMSA or REMSA probe. They're small. And yet we use those probes to do affinity measurements. It seems like there's high affinity to those short probes, just like DNA binding.
Okay, I think due to the time limit, I can only take one more question from Shihan. Hi, hello, Shihan. Have you tried to express the Q1 cDNA, which does not have the intrinsic long non-coding RNA, and see whether it is similar to the binding mutant?
Yeah, you know, we haven't done that. You mean just the cDNA or the whole primary transcript? I'm sorry, what was the question? The primary transcript, I assume. Yes. No.
It's a Q1 cDNA.
Or cDNA.
Yeah.
Yeah, that we haven't done. I would predict it would not bind, but we haven't done that particular experiment. Because, you know, when we did the RIP sequencing, you know, we mainly saw it in the intron 4 region where they have that motif. And we used intron 2 as a control actually for those experiments. For what that's the data that we have. I would predict not, but
probably we can take one more question because we start a little bit late. It looks like the transactivation domain is rich in proline and glutamine, amino acids that are features of protein that do phase separate. Do you think that the phase separation is involved in the binding then?
Who asked that question?
Sarah.
Okay, Sarah, you know, we did experiments with Rick Young's lab, John Henninger, actually, where we took the wild type and the mutant, and we tried to see, could we see any difference by phase? You know, I'm laughing a little bit because, well, you know, sometimes I like to make fun of things for no good reason, but you know, you're just staining with stuff, and it sounds to me like Peter Fraser's transcription factories. You know, it's just a different way of looking at collections of things in the nucleus. Anyway, the sad news was we didn't see a difference from what the staining that we did. That doesn't mean it doesn't affect structures in the nucleus, but at least by that assay, we did try it. You know, we had to make the story sexier by doing phase. I know we failed. Maybe we did it the wrong way, but actually Rick's lab did it, and they have a lot of experience. So, you know, we probably couldn't do better ourselves.
Okay, thanks all for asking the question. So if anyone didn't have time to ask your question, maybe you can email Dan and can have some discussion off the line. So thanks, Dan, again. Thank you. So let's move on to the next speaker. So Our today's next speaker is Dr. Wan Yue, a principal investigator from Genome Institute of Singapore. Yue developed the first high throughput methods for probing RNA structure genome-wide during her PhD years in Howard Chen's lab. After her established her own team at GIS, Yue is very interested to study functional RNA structures and understand their role in regulating cellular biology. Yue has obtained a lot of academic awards, including EMBO Young Investigatorship, the Young Science Award, and the Singapore National Fellowship, etc. I think today she will be sharing her very interesting work about how to map the RNA structures in single cell. Over to you, Yue.
Thank you, Polly, for the wonderful introduction, and also thanks the organizer as well as Polly for inviting me to give this talk, and for Dan for giving a wonderful talk. So I'm Yue from the Genome Institute of Singapore, and my lab has been really interested in developing new methods to look at RNA structure. And so today I'd like to share with you an unpublished work whereby we are mapping RNA structure in single cells. Okay. So as we all know, RNA holds information in its shape, right? So beyond just a linear sequence, RNA, very similar to protein, can actually fold into complex secondary and tertiary structures, which is important for this RNA to function. And traditionally, because of all of the challenges in probing RNA, in obtaining high throughput, in obtaining high resolution RNA structures, for example, using crystallography or cryo-EM, people have now moved to use biochemical strategies. So like chemical probes or enzymes that can recognize double or single stranded regions along an RNA and then actually cleave them. And in more recent years, so traditionally these cleavage sites are actually recognized by sequencing gels. When you run long sequencing gels to tell you where your reverse transcriptase stop. In recent years, we and others have coupled these biochemical strategies with deep sequencing, high throughput sequencing, to be able to obtain RNA structures for thousands of RNAs at one time. And this strategy has been really useful, and people in the field now, it has been about a decade or more, and people now in the field have developed new chemistries, new library preparation methods to look at different transcriptomes, RNA structures during different cellular processes. And we and others have also developed new methods to not only look at, whether a base is paired or unpaired, but also who is paired with who. And this provides, intramolecular interaction information on where along an RNA is interacting within the cell. And it can be very long RNA interactions. They are very far apart, which is traditionally difficult to capture by prediction or experimental strategies locally. And these, pairwise RNA interactions can also be used to look at RNAs signaling, so which RNA is binding to another RNA intermolecularly. So additionally, besides using Illumina sequencing, second generation sequencing, we have also leveraged on recent developments in third generation sequencing, such as nanopore direct RNA sequencing, to actually read out RNA modifications using the nanopore directly. And this has advantages in enabling us to look at isoform specific structures because of the long read nature of nanopore. And so this enables us to understand whether there are structural features along individual RNAs that impact its regulation, isoform specific regulation. And then we have taken these technologies to apply them to biological problems. So for us, we have been looking at RNA aptamers to identify RNA aptamers that can bind and respond to different metabolites in both bacteria as well as eukaryotic Candida albicans, pathogenic fungi, as well as we applied it to look at RNA viruses and how RNA viruses such as dengue, Zika, and SARS-CoV-2 can utilize its genome for infectivity and regulation. And we also apply this into mammalian systems to try to understand RNA structure dynamics, as well as how they can be regulated by different RNA binding proteins during the neuronal differentiation process. So as we all know, traditionally, RNA structure probing is requires millions of cells, actually about 10 million cells, 10 to the power of 7. And so what you are getting is an aggregate of RNA structure information across all of these millions of cells. And recent developments in single cell sequencing has shown us that different cells can have very different transcript identity or transcript levels, and you can understand the identity of each cells much more as you look at different layers of information that's present in individual cells. And so we became really curious to see, okay, besides transcript abundance, do we also see RNA structure differences between individual cells? And that is because RNA structure is involved in so many different cellular processes, from translation to decay to mRNA localization, some of which is difficult to measure in single cells. And so we wanted to essentially answer this question about whether could we add RNA structure information onto some of the other transcriptomics information, and whether this addition to this multi-omic data will enable us to understand the cell's identity much better, just like, for example, in this case, the picture of this cat, which is my cat, and the cells, right, as you get information on DNA sequence, epigenetic information, RNA abundance, processing. Now, if we add on RNA structure, do we better understand the cell? Okay, so to do this, we leverage on some of the structure mapping strategies as well as single cell strategies to be able to do this. So very briefly, we add in this compound that's able to penetrate into cells to modify single-stranded regions along the RNA. And then we isolate individual cells, and we use reverse transcriptase to detect the marked sites. So the way that the reverse transcriptase detects that is as it jumps over a marked site, it actually creates an error. And you can count the number of errors on the site, and we call it mutational mapping, to give you an idea of whether a base is likely to be double or single-stranded. And as the RT enzyme jumps through the site and reaches the end of the RNA, it then undergo a template switching, and then we do PCR amplification, segmentation, and then we sequencing, and then we calculate the mutation size, and that gives us an idea of where the single-stranded regions are located along an RNA. So one of the challenges that we had in developing this protocol was that when you look at millions of cells, you have millions of copies of, let's say, actin or a particular RNA of interest. And so even if your modification rate is extremely low, as long as a very small proportion of it is modified, you're able to jump through capture, and with deep sequencing, you're able to read out which bases are modified. But at a single cell level, you really only have thousands of transcripts of a particular RNA, right? And that is an abundant RNA. And so if your modification rates are very, very low, you may not even get a single modification in the RNA, making it very difficult to measure RNA mutational frequencies well in single cells. And so we needed to increase the modification rates of these RNA, the jump-through rates. And so we tested a combination of different chemical probes plus RT reverse transcriptase conditions to find one. So in this case, the last one, 50 millimolar sodium NAI entry azi plus superscript 2, that actually in the presence of manganese will allow us to have a high jump-through rate. And then we looked at, this is tetrahydrogenate ribozyme. We look at accuracy of the jump-through rate, and you can see all of the methods give you pretty accurate jump-through rates, but with that combination of chemical compound with reverse transcriptase, it gives us the highest frequency of jump-through rate, which was needed for our single cell method. And then the other challenge that we came about was during reverse transcription. Usually the reverse transcriptase reaches the end and then do a template switching. But in our case, because when you have a modification along RNA, the reverse transcriptase not only jumps through, but also a fraction of time it just drop off. And then so there are much fewer cases of the reverse transcriptase actually being able to make it to the end of the strand. And then so our library prep efficiencies were actually very inefficient. And so to overcome that problem, we did a very gentle fragmentation, whereby the average size of the RNA fragments is about 1,000 bases. And then so this allows us to do this library preparation a lot more efficiently. As you can see here, after fragmentation, we can make the libraries a lot more efficiently, whether it is at a single cell level or at 10 cell level, and enabling us to be able to read out at the single cell level. Okay. And then so because we fragment to about 1,000 bases, the end result was that we capture structural information for about 1,000 bases from the 3' end. So when a transcript is pretty short, we are able to capture end-to-end. So from 3' UTR coding region to the 5' UTR. And you can see for the shorter transcripts, we can capture the majority of the transcript. When an RNA is pretty long, to about 2 kb, 3 kb, 5 kb, etc., we are essentially capturing about the 1 kb from the 3' end of the transcript. And the other question that we had was, how deep of a sequencing do we actually need to do to be able to capture RNA structure information? And then so to that, to answer that question, we did a subsampling of the number of reads and to determine, at different read depth, whether it is 2 million, 5 million, 10 million, etc., what is the number of transcripts that we can actually obtain structure information for. So for about 2 million, we can obtain structure information for about 1,000 genes. Okay. So when we did this method, one of the first things that we needed to check was that we're able to capture RNA structure information accurately. And to do that, we perform RNA structure probing in the tens of millions of cells, as was traditionally performed, as well as in 100 cells, in 10 cells, as well as in single cells. And you can see here, this is one example of a highly abundant transcript, RPS27. And you can see that the structure profile across the different scales actually look pretty similar to each other, indicating that our RNA structure probing strategy is actually fairly accurate. Okay. And then one of the best examples that we have that we actually know the RNA structure information on is actually the ribosome, right? The ribosomal RNA. And so we also wanted to look to make sure that our RNA structure probing strategy is actually accurate on the ribosomal RNA secondary structure. So you can see here on the left, our bulk as well as pseudo-bulk reactivity at the single cell level. We get decent AUC, ROC similar to other high throughput RNA structure probing strategies. And in the middle panel, what you have is individual single cell AUC, ROCs. And on the right, what you have is our reactivity, the pseudo-bulk of our reactivity plotted onto the secondary structure of the 18S ribosomal RNA. And you can see most of the red regions indicating they're highly reactive and hence single-stranded occurs on loop regions indicating that we are capturing single-stranded as expected. But the other worry that we had was, okay, so even if you can capture single or double-stranded regions at the single cell level, could you actually capture dynamics when two cells have different structures? Could you actually capture that? And so to do that, we transfected in 3 different controls, whereby we mutated a stem region along an RNA to open it up. So in this case, you're seeing an example of the tetrahydrogen ribozyme, whereby this is the wild type structure, but what we did was we actually mutated these 3 bases in this helix to open up and make it single-stranded. And what we wanted to see is when we make these mutations and we transfect in to one population of cells, the wild type RNA, and into the other population of cells, mutant RNA, and we actually now perform single cell RNA structure probing, can we re-identify the cells according to the structure as what we had actually transfected into the cells? And so indeed we can. So at the pseudo-bulk level, we can detect the structure differences, right? And then when we classify the cells according to the structure differences, we actually identify these 2 populations separate out, whereby one population of cells we actually transfected in the wild type, and then they separate from the population of cells that were transfected into mutant, indicating that we can detect not only RNA structure information at single cell level, but also differences, structural differences at the single cell level. Okay, So now that we have seen that information, we can now look into the genes inside individual cells and ask, okay, which transcript is likely to be very homogeneous, structurally homogeneous, and which transcript is likely to be structurally heterogeneous, whereby depending on which cell you sample it, its structure can look very different. And we have an example here of a homogeneous RNA that looks very similar, no matter which cell you sample the structure from, as well as a very heterogeneous example. And we can then plot the density plot whereby all of these homogeneous and heterogeneous RNAs are plotted on this distribution. And you can see here on the homogeneous front, we do have the positive control 18S ribosomal RNA and some of the other RNAs that are actually associated with translation. And what was interesting to us was that even though 18S ribosomal RNA is generally very homogeneous, we observed this region near the 3' end of 18S ribosomal RNA that's actually very heterogeneous in our data, no matter which strategy we use to calculate heterogeneity. And we realized that this region actually falls into the helix 44. And this is actually very close to the mRNA tunnel, whereby we think that this region is actually involved in direct pairing with the mRNA during translation. And so to test that, to test that, we actually perform an indirect analysis whereby we know that we're able to sort out single cells according to the cell cycle state by certain cell cycle markers. And we know that at different cell cycle states, they have different translation rates. So we basically sorted cells according to whether they're in S phase or G2N phase. And we realized that majority of the heterogeneity again occurs when the cells are in different phases. And this is associated with changes in translation, supporting our hypothesis that we think that there is some translation binding of mRNA that's going on that's resulting in this heterogeneity. So where are these structurally homogeneous regions or heterogeneous regions along the transcriptome? So when we looked at the different windows, we realized that, structural homogeneity, the homogeneous regions tend to be lower in reactivity, indicating that they are actually more structured. And also they are associated with increase in GC content, again, agreeing that they are more structured. And when we looked at along the metagene analysis, interestingly, we saw that actually the more heterogeneous regions, the regions that change a lot, happens actually in the 3' UTR, and the more homogeneous regions are enriched in the coding region. And so then we were wondering, okay, so if these heterogeneous regions are occurring in the 3' UTR, what could be regulating them? And we then looked into RNA binding proteins and whether they could play a role in this. Okay. So when we first did the analysis, you can see here that what we did was we looked for RNA binding proteins that enriched for either single or double-stranded regions or heterogeneous and homogeneous regions. And at first glance, you can see that the correlation is actually pretty good, whereby most of the RNA binding proteins that enriched for single-stranded regions are also very heterogeneous, and that enriched for double-stranded regions are also very homogeneous. And this kind of agrees with our previous observation, whereby the structured elements tend to be very homogeneous, and so there's nothing very surprising. But then when we looked at the RBPs, actually when we intersected the RBPs that enrich for structural heterogeneity versus reactivity, we realized that actually there's a population of RNA binding proteins that are enriched in both, but there are also RNA binding proteins that are enriched only in heterogeneous regions and reactivity on its own. So the correlation is actually not perfect. So then we looked into a little bit deeper to see whether structural heterogeneity could inform RBP usage as well as cell fates, gene expression fates. So we look into PALM2 a little bit more because this is an RBP that has very well-known single-strand binding motif, and it regulates, transcript stability of its targets. So we could sort single cells according to whether they have a lot of PALM2 or very little PALM2. And then you can see here that when we separate the cells according to when they have a lot of PALM2, the PALM2 target sites the transcripts that have PUM2 binding in the cells with a lot of PUM2 tend to be a lot more accessible, single-stranded, whereas those target sites in cells with very little PUM2 tend to be less single-stranded. And this agrees with the understanding in the literature, whereby upon PUM2 binding, the target size actually becomes more accessible. And we also observe that upon overexpression of PUM2, we tend to actually see an increase in heterogeneity in the targets, whereas when we knock down PUM2, we tend to see an increase in homogeneity, again agreeing with, in general, the single-strandedness and PUM2 heterogeneity of its targets. Okay. But we looked at it a little bit deeper. So if you separate PUM2 motifs into whether they're single or double-stranded, you can see that the single-stranded regions tend to have more real PUM2 binding by eclipse. And this is expected because single-stranded PUM2 is a single-strand binder, right? But interestingly, when we separate according to homogeneous or heterogeneous regions, independent of whether they're double or single-stranded, you can see that the trend is actually slightly stronger, whereby the heterogeneous regions, again, tend to be enriched way more in real PUM2 binding sites than homogeneous regions. And when we add, in single-stranded regions, they are more heterogeneous, you can see the highest proportion of PUM2 binding, whereas when you have single-stranded regions, they are homogeneous, you have 0 PUM2. So you can pretty much eliminate the false PUM2 binding sites using heterogeneity. And then when we look at what happens to the fates of these PUM2 target transcripts, if you just look at reactivity alone, whether they change reactivity in high PUM2 versus low PUM2, you can see that between human embryonic stem cells and neuronal precursor cells, that the half-life between the two, you can see that upon PUM2, high PUM2 in human neuronal stem cells, the half-life decreases a little bit, but it's actually not significant if I just look at reactivity, single or double-stranded regions. And if I look at heterogeneous versus homogeneous changes, you can actually see now it's beginning to become significant. And if we incorporate both single and double-stranded information, as well as heterogeneity, now we can actually separate out and show that the PUM2 targets that show both reactivity change and heterogeneous change upon PUM2 binding are actually much more different from each other. They are actually repressed. They have a shorter half-life during differentiation in the presence of PUM2. So we also, in addition to PUM2, we also identify a couple of other RNA binding proteins, whereby if we incorporate heterogeneity information, they actually inform us more on the gene regulation of the transcripts. Okay, so then moving on. So previously we had done this experiment whereby we're looking at RNA structure regulation during neuronal differentiation. And in that study, we had identified many structural changes during neuronal differentiation that was regulated by RNA binding protein. And we were curious whether we're able to, and because this is a neuronal differentiation process that's 14 days long, right? Some of the cellular states are actually quite similar to each other, especially NPCs and immature neurons, which is just a day apart. So we were actually at the single cell level, it has been difficult to separate them out very well just by gene expression alone. So we were also curious to know if we had add on RNA structure information, whether we can separate cellular populations better. And then, so one of the first things that we needed to check was, are the RNA structures equally heterogeneous or homogeneous during neuronal differentiation? And so what we observed was that during human embryonic stem cells, actually the transcripts are way more homogeneous between individual cells, whereby upon differentiation, they actually become more heterogeneous. And we see an example of that in this gene called RPL41, which actually takes on 2 different structures. And during neuronal differentiation, one structure actually becomes less in abundance, whereas another structure becomes more dominant. And we're actually now trying to see whether we can mutate this to understand the function of this change in dominance and structure. But what we are curious about also is if some of these structures are becoming less dominant and there are more dominant structures, can the addition of structural information better separate cellular populations. And so we perform this multi-omics factor analysis called MOFA, and whereby this is an example of just using gene expression alone to separate out different populations. We have 2 batches. You can see some batch effects, a little bit of batch effects, as well as separate across the different stages. And we can separate out using gene expression, except it's not very good. And when we incorporate alternative splicing into the separation improves a little bit, but it's still not very good. And when we incorporate structure information now on top of gene expression and alternative splicing, now we can better separate the different populations. So this indicates to us that RNA structure information can add on to the other multi-omics data to better define cellular identities and separate cellular populations. So I hope that with this talk, that that RNA structure, I can convince you guys that RNA structure information is useful, not only at the bulk level, but also at the single cell level, and that the heterogeneity that we capture between individual cells can yield biological insights in terms of RBP binding usage, as well as gene regulation for these transcripts downstream. And I think at this moment, because it's still a fairly simple system, and I think in future with more complex systems such as tumors and other types of disease models, we'll be able to see the power of that even better. So with that, I'd like to thank you for the time and attention. The work is done by very talented postdocs, experimental as well as computational postdocs, and we are hiring, as well as GIS is hiring RNA PIs. And so I'd like to thank my funding sources and you for your attention. Thank you.
Thanks, you for the very nice talk. While waiting for the audience to raise their question, maybe I can start first. Maybe a naive question. I'm just curious, when you do all this RNA structure probing or analysis, have you ever tried to look at the nuclear fraction and the cytoplasmic fraction? Because I'm always thinking after the intron being removed, maybe the structure, when the RNA transport out of the nucleus, maybe the structure will be completely different in terms of the
so people, Cliff Dang, for example, have done it at the bulk level. So looking at RNA structure changes in the nucleus as well as in the cytosol, and then in the chromatin. So with that, he has seen structure differences as you migrate. So I think that is interesting. At the single cell level, we'll need to fractionate at the single cell level. So I'm not sure. I think people have done single nucleus. So maybe it is useful to look at single nucleus structure versus whole cell. Not sure how. I will need some time to think about it at the single cell level, how to separate it from the cytoplasm from the nucleus. Yeah.
Okay, there's more question coming. So there's one question from Olivier. Is the single cell RNA structure average of all the RNA molecules within this cell? Yeah. If so, is the RNA structure heterogeneity between different cells are expect to be larger than the structure heterogeneity of different RNA molecules within a single cell?
Okay, so this is a complicated question. So let me see. So yes, so the first answer is yes. So when you measure currently RNA structure in a single cell, we are looking at an average of all of the RNA molecules, of the structure of all of the RNA molecules. For example, actin, 1,000 copies of actin. It is an average of that in single cell. And there has been really beautiful work done by Sylvie Ruskin as well as Danny Incarnado on single molecule heterogeneity. And I think that is really interesting. And it's something that we're looking at also. I think we will have to, try to incorporate that at the single cell level. And then I think whether the heterogeneity larger than of different molecules, heterogeneity between different cells to be larger than heterogeneity of different molecules. We currently the work that has been done is this work is done in single cell, right, which is the average of RNA structures in a single cell. or it is done in bulk but single molecule. So then that is different structural populations of the same RNA, but average across all of the cells. So I think unless we do single molecule, single cell, it is difficult to know which one is more heterogeneous. Means it's difficult to know whether the variation is greater between individual cells or between different molecules of the same RNA. And I think, yeah, that waits to be seen. Yeah.
Thank you. I think due to the time limit, I will probably take one or two more short questions. So Chen Hao asking, does the gene expression affect RNA structure? It seems the homogeneous structure gene, structure genes have higher expression level.
Does the genes, homogeneous structure genes have higher?
Has higher
We
expression.
actually didn't see that correlation between, I think we plotted that. We did not see a correlation between structure and expression. Yeah, I don't think, although because of the method on the sequencing depth, we do tend to capture some of the more abundant transcripts. So in this case, we capture the top 1,000 transcripts. So I cannot rule out. So maybe in that top 1,000 transcripts, because they're all fairly abundant, so we don't see the correlation. So that I cannot rule out. But as of right now, within the 1,000 transcripts, we do not see a correlation between heterogeneity and abundance. Yeah.
Oh, Shoha was asking the heterogeneity that you see in the single cell level. Is it stable over time?
Is it stable over time? Depends on what you mean. So, for example, if we were to, for example, the simplest thing, right? If we were to transfect in an RNA of known structure, for example, tetrahymena or dengue or whatever it is, right? And we map it, it is very reproducible. So it is stable, but I think because the cells are undergoing all kinds of changes, and we think that a lot of the RNA binding proteins, for example, the abundance change during differentiation or during stress. So we do see quite a lot of variation. But if we transfect in an RNA into a cell and we test it multiple times, it is stable. Yeah.
Okay, thank you again. Yeah, apologize for those that I didn't manage to read out your questions. So we will be sending your question to Yue, and then she can follow up with you probably via email. Okay, thank you again for Yue for sharing your work. Yeah, thank you.
Thank you. See you.
Thanks everyone for attending today's seminar. Thank you so much.